And whenever you come, you take away some good ideas. So all PG students probably they are lucky enough that you are in the department, and particularly I talk about entomology department. It has been a leading department right through establishment of DNA itself. So as Dr. Srinivasan told, my present background is extensive. Away from active research, but I work with fruit flies, and I'm still continuing some of the aspects, but probably more relevant to the applied aspects rather than the theoretical aspects. Most of you might be well adversant with this pest, fruit flies. Very nostalgic pest. Insects as a whole, we know that they are the most dominant clade among all the biodiversity, constituting about half of the total diversity. And Diptera, to which these fruit flies belong, they again are one of the dominant order in the insects itself. Why insects are so dominant? There are many reasons you might have been taught. Why they have evolved such beautifully to almost every climate, every location, every host. But now it is quite common and has been reported by many workers that one of the main reasons behind dominance of these insects is their association with microorganisms also. And these microorganisms, they do, some are obligate symbiotic bacteria, some are facultative, and some may be playing some other roles. But definitely, these symbiotic bacteria, they have a very, very important role in the dominance of insects. This is just an overview of the fruit flies. This is a very brief lecture. I'll just be giving you some of the ideas what has been done in symbiosis in insects in general and fruit flies in particular, because this family, Tephritids and Pentatomidae, these are the two families which have been exploited to a great extent among the insects also for study of insects. Hemiptera, particularly because they feed on liquid diets. And these diets, they are deprived of many nutrients, as we all know. And these microbes, these symbiotic bacteria, which are associated with these hemipteran insects, they help these sap-sucking insects to fulfill the requirement of their diets, whether they are the vitamins, minerals, or any other thing. So they are very important in the sucking insects. And in fruit flies, if I talk about fruit flies, they are the most nostalgic pests, causing an estimated damage of around 8,000 crores in our country annually. Maybe not severe in this part of our country, but when you go to the north, western plain zone, northeastern zone, or northern India, these fruit flies, they cause havocs, particularly in fruits and vegetables. As we know that they belong to superfamily Tephritidae and family is Tephritidae, but more than 5,000 species they have been described till date in our country. From about 500 genera, these are some of the important fruit flies across the bird, like Ceratophrys capitatis there is there, Regulatus is there, Decus is there, Anastropha is there, and Jugodacus is there. Jugodacus you might be confusing. Earlier it was named as Bactrocera. Most of the fruit flies, they were prevalent in our country. They were nomenclated as Bactrocera mostly, but recently researchers, particularly Clark and his group in Australia, they have studied extensively this Jugodacus complex, and they have now upgraded this Jugodacus as a genre, though not confirmed by many workers, but still it has been reported and it has been well apprehension also by many workers that this subgenus Jugodacus, it should be considered as a separate genre comprising many important species in our country, like Jugodacus cucurbiti, which is an important pest and cucurbit. Jugodacus tau is there, and Jugodacus scutularis is there. So these are some of the changes which the taxonomists, they are keep on doing, because many new species, they are being discovered, many new clades are being discovered, many cryptic species, they have been clubbed into a common genre. So many taxonomic changes are in progress, and we keep on updating them. These are the most nostalgic pests. Why? Because they are the pests of fruits and vegetables, and mostly these fruits and vegetables, they are eaten raw. They are not cooked. So the life cycle itself is explaining why they are nostalgic or very difficult to control, because the fruit flies, they lay eggs inside the fruits or inside the host tissue, and we cannot overdose these products with insecticides, because that will carry over the residues of insecticides, and this may be harmful to the consumer also. There have been many problems with excessive use of insecticides, particularly in case of these fruit flies. Even sometimes consignments costing more than thousands of crores, they have been rejected by some countries only because of heavy residues on these fruit consignments. Particularly mango is one of the example where some consignments, they were destroyed by these importing countries. Fruit flies, as I told you, they lay eggs inside the fruit. So generally the immature stages, they are concealed inside the fruit. Even heavy doses of insecticides are also not enough to take care of these pests. So a consortium of approaches have been deployed for the management of fruit flies, including MAT, that is male annihilation technique. If anything, what I'm saying is not clear, you can pinpoint, or you can raise the query at the same time. MAT is there, but which has been employed to a great extent. MAT is nothing but it is a male annihilation technique, because we know that in insects, parapheromones, which are also called pseudopheromones, like Culior, methyluginol, and many others have been invented now, which are being used to capture the male fruit flies, and they are the best tactic not only to mass trap but also for the monitoring of these fruit flies. Apart from this bait application, that is some sort of lucrative material, trapping material involving jaggery or anything else with insecticide, that is also a common strategy. But apart from this, many advanced countries, they have now gone for bagging, because no other approach was working for the management of these pests. So they have now started bagging of individual fruits, so that the fruit flies is not able to lay eggs. So uh, briefly, I must say that these are the most nostalgic due to their a specific type of life history or biology. If you look at the bird distribution of these fruit flies, there are the different regions, geographical regions, like Indo-Malayalam region is there, Australasian region is there, and two things are important. I'll not go into detail of this, but two things you must focus upon. That if you look at these specific regions, we will find there are different type of species prevalent in these regions. Though this Indo-Malayalam and Australasian region, it is the dominant as about if you look at the number of species, there are more than 1700 species which have been described from these two regions itself. But if you look at the Arctic region, uh, then neotropical region, there are the specific type of fruit flies. If you look at Indo-Australian region, there is specific type of fruit fly group. So it means these fruit flies, they have adapted to specific climates and they are restricted into that specific region now. But as we know today that invasion of species across these regions is one of the key things that many countries they are taking care. Apart from, except India, I think. Our quarantine system is not that strong. But if you go to Australia, Europe, their quarantine system is very strong and they do not allow entry of any material or any pest into their system. Many invasive pests they have entered into India. There are many reports, right from this Courtney Coulson scale to this tomato pin bum, fall army bum. These are the recent examples which are now causing havoc in this maize and solanaceous crops. So our quarantine system is not that strong. So to avoid the entry of these specific fruit flies from one region to the other region, it is the most important because this regulated species particularly, it is a dominant pest of apple. Ceratitis, it is a very dominant and very devastating pest of citrus industry. So though not official, but unofficial, I am telling that we have encountered fruit flies in apple also, in pomegranate also, in kiwi fruit also. But though we could not report these invasions only because of once they are reported, whole of the export industry of apple and these fruits they will be come into question. So very challenging task is to avoid entry of such type of species into our country. Some of the Indian fruit fly, if you look at the diversity of Indian fruit flies, more than 400 species already reported and the number is increasing. I'll show you two more species. They have been added from our place itself. So this number is keep on increasing with every day, every week. Some 80 genera have been reported in the country. Coming to fruit crops, these are some of the important, not all. I have mentioned only three, four species which are more devastating and very important, like this Bactrocera dorsalis. This is a pest of mango, guava, sapota, lychee, and many other fruits in India. It was first reported by Handel in 1912 from Taiwan. Though it has originated from Asia, but the exact place of origin is still a matter of question. Some workers they say that it originated in China, some say originated in Southeast Asia, but some other reports are there which say that this species has originated in India. But still, not a common acceptance among the workers. In India, it was first reported by Beji from Bihar. Most of the fruit flies, you will see that they have been reported from Bihar. Uh, but it may be because of but my assumption is that Pusa Institute was in Bihar and many of the workers, they were stationed around that entomologists. So they were tapping those locations also. Otherwise, it may be some other locations also. 450 host plants have already been reported. And it's a complex of species. Many cryptic species are there. Recently, this is a report by Clark, Mark, and co-workers who have studied all these cryptic species, and Earlier, about six cryptic species were there which were described in this clade Bactrocera dorsalis, but now because they can intercross, interbreed, so now they have clubbed these species into a single species, that is Bactrocera dorsalis. Like in Vedans was reported as a separate species, Papai was reported as a separate species, but because you know what is the difference between a species, it should not interbreed among themselves. So these species they can interbreed among themselves. So that's why they have been clubbed together. Bactrocera jonata, again very important, very serious pest in our country. It was reported again by from Bihar, I think, in India, but definitely it is of Indian origin. 1842, it was reported from Bengal, uh, and it is well established in many South Asian countries. Egypt, it is well established, but it has been reported that it can establish to many other locations in the southeastern region also. Bactrocera correcta, though recently uh, noticed as a serious pest in fruit crops, but it was reported again from Bihar by Bej in 1916, widely distributed in South Asia and Southeast Asia, detected in China also, and highly adaptable to new climatic conditions, enabling it to spread rapidly in many other countries also. 62 host plants, they have already been uh, noticed as a host of this species. As far as this fruit crop, this is a map which you can see and just get an idea. Almost every part of the country, you can see these three species, though uh, the density is somewhere more and somewhere less, but overall, if you see this map, almost everywhere you can find the reports of occurrence of these fruit fly species from across the country. Some apart from this, I've already told many new species they have been encountered in the fruit crops also. Like regulatus has also been reported in our country, and it is a very serious pest of particularly temperate fruits, apple, and then dorsalis has been found to damage this pomegranate and kiwi fruit also. Uh, Coming to vegetable fruits, these are some of the important species, three, four species I'll mention, but there are a number of some 20 to 25 species are there which have already been described from our country, which are causing damage to various extent in our country. But this is the most important. Earlier it was Vectrocera cucurbitae, but now it is renamed as Geogodicus cucurbitae. It was first reported again. If you see, it is from Bihar, but the reason I have told you, it is from Indian origin. This is the bug we have carried, and we have assigned India as the origin place of this fruit fly species. It is widely distributed in Asia and Africa, highly invasive and adaptable to new climates. This species, only because of this character, it has established many locations all over the country and away from the country also. 125 host plants, and the major pests are the cucurbit crops. If you see the distribution, I've given a map showing the diversity of this particular species. Earlier, this was a very dominant species in our country also. When I did my PhD in 90s, this was the most dominant species in our place. But after 15 years, when we restudied the same location, this species was overpowered by another species, that is Vectrocera tau, which was earlier a suppressive species, now that has emerged as a dominant species. This you can see based on MTCO1 gene, mitochondrial cytochrome oxidase 1 gene. We have studied different populations from across the country, and some other workers also they have reported the populations of fruit flies from different Asian regions, some from other regions also. So we can see that all the populations, they are from the single haplotype. So there is not much variation in different species, and based on this work also from the various different parts of the country, whether it's the northeastern region, north hill region, northwestern plain region or other, we got a single clade and a single haplotype. That's why we designated that this probably has originated from India. Uh, Pumpkin fruit fly, I was telling you this species earlier was not a dominant one, but now it is particularly causing a serious loss to cucurbitaceous crops, and it's a major pest. 8 cryptic species in B-tau complex has been described by Janglok and Barkas, widely distributed in Asia. China also, it was a very serious pest, and it was reported from China itself in 1849. This is the species complex of this Jugodakas tau. If we see the work of the Jamlonglik 2003, they have described different groups of B-tau complex, some 7 or 8 subgroups they have assigned, but during our study, we could find that from India, when different populations were studied, they matched to a single group of, as described by the earlier workers, and all the Indian population that was probably it falls into a single clade. This is Scotellaris, again a new species described from our location. Maybe it's a serious pest because of its slightly different type of nature of damage. Other species, I've told you that they females they oviposit their eggs in fruits or other parts, but this species it is known to cause damage to the tender shoots also. It lay its eggs. So it lay its eggs on the tender shoots or very small fruits. And generally, most of the other species, they lay eggs more than 20-25 eggs at a single location. But this Scutellary species, it lay its eggs singly. So its damage potential is more vigorous than the other species only because of this slightly different nature of oviposition, because it can lay up to 150 to 250 eggs. Of if it start laying eggs singly, a single female can destroy more than 100 fruits simultaneously, which in other species is not more than 5 or 6 fruits. This is the distribution of various fruit fly species, Petrocera taus, Cotellaris, cucurbitae, in the country. And again, you can see almost all parts of the country, they are having some or the other species. I was telling you some of the new species. This was earlier misidentified as Bactrocera femoralis, but now it has been reported that and the new name has been given on the name of one of our colleague scientist, Dr. Devendra. Its species has been named as Dr. Bactrocera devendra, and another species. This is Bactrocera prabhakare. You might be looking at some of the work. He was my student, so this species has been named on his name. This is again a new species. It was reared from some wild fruits. Solanum khasianum is a wild fruit from which this fruit fly was reared. It was coming in a bigger way uh, in many locations, but exact uh, nature of its origin, location, its biology was not known. So then we came to know that this is from this particular fruit. Coming to this particular aspect, symbiotic association, I was telling you that one of the reason for dominance of insect pests is their association with microorganisms. And these microorganisms, they are playing a number of roles. And as our science understanding of these associations is advancing, many new roles of these symbionts, they are being assigned in the dominance of insects in their most adjustable, most adaptable in species. Probably these insects, they play a very useful and very diverse things they are playing. If you look at the dominance of these symbiotic bacteria and insects, upper one you can see These black lines are there, they are dominating. So they are the different species of insects where you can see this particular group of bacteria that belong to Proteobacter. They are the most abundant and most dominant group of symbionts which are associated in insects, except one species, that is termite, because termites they have a different sort of mechanism. So there, the spirochetes and in some other species, firmicules, they are the slightly dominating. But as a whole, if you look at this bar diagram, we can clearly assign that most of the symbiotic bacteria, about 68%, 65%, they are from Proteobacteria group itself. And within this group also, particularly to family Enterobacteriaceae, and most of the dominant genera, if you talk about it, is the Klebsiella is there, Pentoa is there, Enterobacter is there. Some of these are the most dominant ones. This is the digestive tract of different insects. You can, wherever you see these blue regions, they are the midgut regions, and some black spots are there. So the black spots in a particular region, they are depicting the dominance of these symbiotic bacteria in insect groups. Like in this Hemiptera, if you look at this kissing bug, you can see the midgut region is bulged and it is harboring, you can clearly see the black dot. So it is harboring the symbiotic bacteria. These bacterial associations, I have already told you, they can be obligate, they can be facultative, and some may be phytoparasitic also. But to our concern, most if you look at the pest management, most important targets, they are either obligate ones, or secondly we can go for the facultative ones also. Like in Hemiptera, we have got both this obligate and facultative association, and both of these can be targeted. I'll come to that, how we can target it. I'll not detail these things, but if you look at particularly this fruit flies, symbiosis in fruit flies, it is not a new thing. It was described since the beginning of last century by Petri, when when he this Bacteroceras aurea, he described some bacteria-like organisms, masses of this bacteria-like organisms, which can be inherited vertically within this species. And this is a very old report, but still the base of this symbiotic association among the insects. Some of the extracellularly inherited symbiotic bacteria, you can see a number of hospitals. This list is a small one. There is a long list. Particularly this Pentatomidae and Tephritids, I have collected some references. So you can see a number of species. They have been isolated, and some are transmitted. Which have been collected through ovipositional smearing. They are transmitted by the females by ovipositional smearing, and some others, they have been transmitted after laying of the before laying of the eggs. So there are two types of mechanisms how the females, they are transmitting these bacteria into the new generations. Most of the obligate symbiotic bacteria, they are vertically transmitted, and the facultative ones, they are mostly horizontally transmitted. This is a common thing. This is the bacterial pathway inside the fruit flies. If you look at the this oesophageal region, which is the white one, this one, and the rectal pads, they are the most dominant regions in fruit flies which are inhabited by these symbiotic bacteria. These are the roles, like right from orientation of the adults. I'll cite some of the examples, some of the case studies from which you can easily understand how they are playing different roles. Ovipositional behavior, they are manipulating. Then nitrogen fixation, like they can this degrade urea and some other compounds into the nitrogen. in the insects which require nitrogen where the food is lacking in the nitrogen. They provide lipids, proteins, vitamins, insecticide degradation. Many of the insects, they are playing these symbiotic bacteria, they are playing very useful role in degrading these insecticide into the compounds which are less harmful. In some insects like honeybees also there have been association with the symbiotic bacteria, and they can help in selective entrance of some compounds within the peritrophic membrane. They form a sort of this peritrophic complex, the network of these species, and they can selectively allow entrance of certain nutritional components. They also help in mature stages, growth and development. They enhance male fitness in set technique, and more recently they are more prominently used in IIT also. So I'll come to that later. These are some of the, particularly if you look at some of the insects, these are some of the roles, particularly if you look at this pesticide degradation. We know that two harm this jasmonic acid and salicylic acid, they are the most prominent compounds which are working as a plant defense mechanism engine. And these symbiotic bacteria, they can manipulate these enzymes in such a way that they can uh, help the insect to invade these resistant plants. Similarly, in case of pesticides, I've told you that many examples are there. Honeybees also, they are known for these symbiotic associations. They are known for degradation of certain chemicals, insecticides into less harmful ones. They can protect against parasites and predators also. Some of the examples where they supply nutrients, there is a long list. Again, many of the species you can see, they help in secretion of essential amino acids, vitamins, minerals, or other compounds. And this association is only reason why insects, they are capable of feeding on host plants which are even deprived of essential nutrients also. This is the simple example of aphids, which are the sap homopterans, which are sap feeders, and these sap feeders, the diet which they take is generally deprived of many essential nutrients. But symbiotes, they usually supplement their diet, and they are able to meet the requirement of the insect. This is some of the examples where these symbiotic bacteria, they are acting as diluting the plant defense mechanism, or they are confusing the plant to secrete certain enzymes, certain chemicals, so that the insect can easily harbor that particular host. Many examples are there. These are some of the common examples. This you can see here, particularly in case of apple. If you see this cytokinin based release confusion of the host plant, where when these bacterial symbionts, they inhabit the plant surface, they produce specific chlorophylltic enzymes, so that these insect can come and inhabit that particular part of the plant. This is particularly example of confusing the predators and this parasites. Mostly this ladybird beetle is a predator of this aphid, and it is mostly of green color, red color. But when it is inhabited by the number of symbiotic bacteria, it increase in their gut, their color changes to green. So this ladybird beetle usually skip this particular host, and generally it is a sort of mechanism of confusion. Even by manipulating or camouflaging the insect also, these symbiotic bacteria can help the insects. This is pesticide degradation by symbiotes. You can say it's an example of phenitration. It is converted into the compounds which are less harmful to the insect. So the insects which is inhabiting these symbiotic bacteria that can easily resist the insecticides, and this is only one example. There are a number of examples like this. So how we can use these pests using these symbiotic bacteria? The answer lies in whatever I have told you earlier, because they are playing many useful roles. And now, with understanding of their mechanism, how they are working for the benefit of the insects, the same understanding can help us to manage that particular insect. Some of the examples I'll cite, like antibiotics. This is the most commonly used tactic, and a number of antibiotics, they have been used to suppress the population of these symbiotic bacteria. These bacteria, symbiotic bacteria, the mode of action is like when the fruit fly, I take the example, when they visit a particular host, they regurgitate these symbiotic bacteria onto the host surface. And once they multiply on the host phyloplane, the insect or fruit flies get attracted to that particular host. So once you use these antibiotics either on the host plant or on as a uh, food to the insect, we can mix these symbiotic bacteria with some antibiotics and make them as an insecticide also. There are studies like that also. Like I told you, these are the dominant, Pseudomonas proteida is dominant, Klebsiella oxytoca is dominant one. So we can mix them with certain antibiotics. We can mix their various formulations with antibiotics and use them as an insecticide. So that way also we can suppress the population of symbiotic bacteria inside the gut of the host plant. And once this they are deprived of these symbiotic bacteria, their normal biological development is hampered. Either they are not able to identify the host, either not they are not able to oviposit properly, and if they are able to do this, the young ones, they are deformed, they are not capable of completing their life cycle properly. So many examples are there. You can see here also that they have been employed successfully, where they can make the uh, this diet of the insect deprived of certain this particular amino acids or vitamins. So once this diet is not balanced, definitely the insect biology will be hampered. Again, some of the active substances you can see here, and how they are working. You can see here that in different insects, when these whether it is copper oxychloride or some other substances, when they are used against these fruit flies, they make the insect deprived of particular either nutrient or their defense mechanism or their normal development. So a number of commercial products are also now available, which can be used as a mean of management against the pest. Schematically, if you see, this is two example, Pentatomide and Tephritids. Again, if you see this Pentatomide, it's mostly in the hindgut region, but in fruit flies, these bacteria, they are inhabited in the esophageal valve and rectal pads. And once they are separated with the antibiotics, their normal development is hampered, their oviposition is hampered, and mostly they are deformed or killed during the course of this application of these antibiotics. These are some of the roles, and they can be utilized, all the roles, whatever these symbionts they are playing in the ecology and physiology of the host insect, they can be of a great use for the management of that particular species. We can alter that role and convert that mechanism as a defense for our uh, crops, like paratransgenesis is there, autolysis is there, many other mechanism, prevention from predators and pathogens is there. So once you alter that mechanism, you can make that particular insect more susceptible to a this predator or a pathogen. So it is mainly the altering, the understanding and altering the mechanism, but the symbiotic bacteria, they are playing inside the gut system. Uh, I've told you that most of these symbiotic associations, they are either horizontally transmitted or they are vertically transmitted. So if they are vertically transmitted, the window is quite open. There are the target window you can see. We can go for making aposymbiosis. We can go for making this biopolysis is there. Aposymbiosis is there, where you can kill or make the insect free of these symbiotic bacteria so that they could not develop normally when they are having a full strength of the symbiotic bacteria. Similarly, in case of horizontally transmitted bacteria, simply this use of antibiotics, because they are mostly transmitted through the host phylloplane. So simply using antibiotics can be a very good window for the management of the insect pest. These are some of the species which have been described particularly in different species of fruit flies. Mostly if you see the species, either they are the Enterobacter, Acinetobacter, then Bacillus is there, uh, Proteus is there. So mostly from different reports, you can clearly see that the species of these symbiotic bacteria, they are almost some or almost common to different locations, whether it is in Asia, Australia, or the other region. Most of them are predominantly from either Enterobacteriaceae family or in the Proteobacteria group. Again, these are the different roles in some other insects. Uh, Coming to this attractancy, one aspect I have told you that they can be managed by using antibiotics. But again, this is another very useful concept, because these fruit flies, they are attracted to the cues and chemicals produced by these symbiotic bacteria. There are some definite cues which these symbiotic bacteria are producing, either attracting the females to oviposit the eggs in that particular fruit, or attracting that particular species to feed on that particular fruit. So there are different cues which are working, and these attractiveness of the symbiotic bacteria, they can be employed as a mechanism of its management. We have evaluated, we have studied different fruit fly, this specific symbiotic bacteria, and different type of responses we have got. And also we have studied that uh, it is only up to 10 to 15 days when this bacterial symbiotic population, after the birth of the adult, it keep grow exponentially, but then it becomes almost static. So up to that particular age, when the insect, particularly if you look at the female, when it has to lay eggs, so it means it must be producing certain chemicals like vitalogenin and oogenesis, etc. So they can be of help for management of these particular species. Here you can see that when the attractiveness of vast bacterial pellets, only three species they were studied, and when the vast bacteria was studied, it was the Pentose agglomerans which was the most attractive to Bactrocera cucurbitae and Bactrocera tau. And within it also, if you look at the scenario, it is the females which are more attractive. Mostly whatever the monitoring and trapping devices we are using against fruit flies, they are targeting only the male. But actually, the damage is being caused by the females. So once we get a compound which is attractive to the female, that can definitely be of more useful for the management of this particular group of insects. Similarly, uh, when this broth culture of these symbiotic bacteria was studied, again, if you see, earlier it was when it was used as a Bast bacteria, it was pentoagglomerans, but when it was broth culture, so it was Klebsiella oxytoca, which was the most attractive. So it means some particular odor, some particular chemicals, they are being produced once we wash the bacteria. Those chemicals were washed, and hence that was the second in attractiveness. And this Klebsiella was the most dominant attractant for these two species. And again, if you see the number of females which were attracted, they are almost similar to the number of males attracted. So it means they are the good candidate of for luring or attracting the females of this particular insect group. Furthering this study, because these were attractive to the fruit flies, so we can go for this GCMS analysis of particular bacterial isolates, the other of bacterial isolates, and we isolated many chemicals when we studied the GCMS chromatograms, but most important one were the phytophylline and oxyl and cardisol and muscalure, which were the most important and somewhat related to the insects. Other compounds may also be related, but though they have not been reported earlier by any worker, which could have any impact on insects. But definitely these three compounds, this we studied during 2010, but now I will show you one paper after 13 years of this work, but we carried. Uh, there are some reports how this caryophyllene compound is helping these fruit flies. At at the time we reported this chemical, we were not understanding the exact mechanism. But now the mechanism of this caryophyllene compound has been solved by some workers, and I'll come to that part later. So these are some based on these attractiveness studies. These are some of the targets where we can use these symbiotic bacteria in applied aspect for the management of the insect, like bacteria based baits we can make either as a Bast bacteria, their pellets, their broth culture, or anything, any compounds you can make, they can be used for mass trapping and monitoring of fruit flies. Bacteria based bait application as a as an attractant and as a killing agent. When you mix these bacteria, attractive bacteria, with some small quantity of insecticide and keep them as a bait station, the fruit flies will be attracted to this particular odor of the bacteria. And because that will be mixed with the insecticide, that can very easily be accepted as a bait station. Exploiting bacterial cues, as I've already told you, we have to understand deeply what are the chemicals, what are the cues, what are the various mechanisms, various pathways of interaction of these symbiotic bacteria with the host plant, because it is not a this bacteria and insect interaction. It is a tritrophic type of interaction where plant uh, then insect and bacteria, they are mutually benefiting or uh, harming each other. So we have to under this tritrophic interaction very precisely, only then we can deploy these associations into a game changer management strategies. Use of transgenic symbionts, again, this was quite old idea. We can uh, make these symbiotic bacteria uh, toxic to the host insect using certain specific genes. Though in our country it probably will not be accepted very easily, but the approaches they have been reported by some workers where we can make these bacteria as a transgenic carrier or carrier of toxin genes because they are attractive, so they are easily accepted. They are residing in the host gut, so they are easily accepted by the insects. So this can also be one of the mechanism, though slightly different from this approach. We know that recently many new technologies with the understanding of gene silencing, gene editing, many new technologies, they are also coming up, like CRISPR is there, RNA is there, RNA interference technologies, interference technology is there. So in IIT also, SIT also, these bacteria, they are very helpful and they are making these techniques more effective in some parts of the birth. This is, but I have already told you that ovipositional aversion, and if the compound I was telling you, this caryophyllene, it was reported by us in 2010, but now there is a report in 23 by some workers that how this compound is uh, being used as a aversion mechanism in Bactocera dorsalis. So this Bactocera dorsalis, they avoid oviposition on the egg-infested fruits 48 hours after initial oviposition. So how it is doing, how the female which is basting a fruit, it comes to know that this fruit is already been laid eggs by another female. So this particular compound caryophyllene. Once fruit fly oviposit eggs into a particular fruit, there are triggers for the release of this particular compound, caryophyllene, and this start emitting. Other fruit fly, when it visits that particular fruit, it comes to know that this fruit has already been laid by another female. So to avoid congestion within a fruit, within a host itself, because we know that in a particular fruit, only 20, 25 larvae they can survive. So this compound is playing a very useful role. How the fruit flies, they are maintaining this particular number of population within the fruit hosts also. Similarly, if you look at another aspect, some sex pheromone production by males in fruit fly. This is also a new vista. This two chemicals, they have been reported by Ran et al. They are the TMP, trimethyl pyrazine, and tetramethyl pyrazine. These two sex pheromones, they have been isolated from the male rectum. And I've already told you that in fruit flies, particularly Tephritidae, this male rectum and also phageal valves, they are the reasons where these bacteria, they are the mostly inhabited. So these mature virgin females, they are strongly attracted to TMP and TTMP. So these two compounds, they can now be utilized for the management of for the attracting the females, which is a big challenge. Some workers, they are working on these aspects. They have isolated some more compounds also, but definitely in the coming time, some female specific lures, if they come into the market, they will be from this symbiotic association of these insects. I was telling you some transgenic compounds. We studied this colonization of marker stain, though it was not transgenic, but we made two bacterial species, this Bacillus subtilis and Pseudomonas potida. We made them doubly resistant to two antibiotics, streptocycline and rifampicin, resistant up to 500 ppm concentration. And when we fed these uh, antibiotic resistant, doubly antibiotic resistant strains to the fruit flies, we can clearly see the status of establishment of these doubly resistant strains into different organs of the fruit fly up to the death of the adult, that is up to 18 or 20 days. And the another one, E. coli, was a marker stain. It was uh, got from USA. This was genetically engineered Marcus 10. So all these species, they easily establish the gut of the fruit fly, which can clearly give us an idea that if we plant an engineered symbiotic bacteria, they can be easily established into the host gut system, and then that can easily be deployed for altering the role it is playing into the host physiology and ecology. Uh, some of the future perspectives and challenges, though there are many challenges, in spite of our understanding the mechanisms, the pathways, uh, improvement in knowledge of our understanding. Still, there are many challenges, though these are the options, but simultaneously, these options are equally clubbed with more serious challenges, because if we talk about using plasmids and transposons to manipulate symbiotic bacteria, how these transposons, bacteriophages, these plasmids, they will behave inside the gut. That understanding, that mechanism is still not very clear to us. Similarly, using this RNase technology, using CRISPR technology. Though idea is there, they can be deployed. Our knowledge of understanding is very good now. We can silence a gene, we can edit a gene, we understand the mechanism. But once we use this technology with symbionts, how it will behave inside the gut, that is also a matter of question. And simultaneously, the cost factor is again a bigger challenge. These techniques now, they look like very lucrative, but they are very costly. Making a RNAi based fruit fly control mechanism, it will cost. It will take, so it is so laborious and so costly. Whether our system will afford or not, that is also a question. Targeting specific genes from host insect via symbiotic mediated techniques, gene silencing or gene editing technologies, again, that is very again a very good option. Manipulation of gut symbionts with CRISPR, this has been rated by the Science magazine as the most futuristic, most noble approach in the coming time against insect pests. Methods such as trial and sit and incompatible insect technique, they have also been made more useful with the help of these symbiotic bacteria, because when we feed these symbiotic bacteria to these sterile insect technique, they become more competitive, they become more proteobactic, and they can easily compete with the natural population of male insects to mate with the females in the host populations. So these symbiotic bacteria, now detailed studies, they are still continuing. We have to understand this mechanism more deeply, more exhaustively. What are the pathways? What are the mechanisms? What are the biochemical changes occurring within the system because of these symbiotic surfactant as a using of antibiotics, using them as a bait station? They are the approaches which can be easily deployed with the simple understanding of the role of these symbiotic bacteria associated not only with the fruit fly, but with other insects also. Many studies, if you see the recent literature on symbiosis, uh, a, a whole number of very good papers, they are coming up nowadays. Recently, in last 10-15 years, the interest on these symbiotic bacteria, that has uh, attracted many, not only the entomologists, but the biotechnologists also, or even the microbiologists also. So you can go across many papers, and if you look at some of the options, these are some of the options which you can for your post PhD or during PhD, you can use this as a vehicle of your thesis also. So with this, I thank you very much. Uh, though this is some of ideas are theoretical, but some can be conceptualized. And if there is any query, you are